Good afternoon, just about, and welcome to our webinar today on IsampleON, or the Invasive Species Awareness and Monitoring Program for Lakes Education Ontario. My name is Terry Reese. I'm the Executive Director of FOCA. Joining me today are my colleagues Michelle Lewin of FOCA, Rebecca Dorzio of the ISC, the Invasive Species Centre, and Colin Casson, also of the ISC, who you'll hear from momentarily. Thanks, everyone, for joining us. We'd like to start this meeting by recognizing and thanking the traditional stewards of the lands and waterways where we work and live. The waterfront lands of Ontario are the traditional territories of many diverse Indigenous peoples who recognize the sacredness of water, the interconnectedness of all life, and the importance of protecting land and water for the generations yet to come. We are all on a journey towards truth and reconciliation and offer this recognition as an important step in that journey. We are joining this virtual meeting from different locations, and I encourage each of you to dedicate yourselves to move forward in a spirit of respect, reciprocity, learning, and sharing that learning. So thank you again for joining us today. Just a couple notes here. We should all be familiar with Zoom, but you're all going to be muted. We've got a number of people here, so just to eliminate and reduce the number of distractions and noise, you'll be muted, but you can comment through the chat feature if you have questions. And any of your questions about the content or the technical stuff can be put in through the chat feature, which you can find by mousing over your screen and clicking on the chat feature, or you can tap if you have an iDevice and the questions will come to us that way. We are going to have questions at the end. So if you think of something during the meeting that's on your mind or that was unclear, please do put them in the chat. This is going to be recorded, so you will get a copy of this webinar, and further instructions are going to follow as well via email related to the technical aspects of the program. But this is meant to be your refresher and your introduction to the program, so hopefully we'll address many of your questions here. So again, I'm Terry Reese. I'm with FOCA. FOCA, just as a reminder, is the waterfront representatives for 525 associations across the province. We've been around 60 years. It's our 60th anniversary. Very proud to represent and educate and connect with so many great interested associations and individuals across the province, located from Kenora to Cornwall. And as you can see on the map, we're covering the coast to coast to coast here pretty much. So we're the voice of Ontario's waterfront, invasive species being a very significant issue for all of us and for FOCA. So that's why we're happy to have you part of this program and this conversation today. The program, the iSample program 3.0, we're calling it, is going to be primarily driven through our offices in Peterborough. So FOCA will be your main contact. ISC is our technical partner here and providing a number of resources to make this program happen. So just so you know the who's who, and we'll have Rebecca and Colin introduced in a moment. Just a reminder how important volunteer citizen scientists are in this whole understanding of our aquatic ecosystems, both in monitoring the systems, being the eyes and ears of all of us. So there's no way to replicate the fact that all of you are out on your lakes year after year for days and days at a time. So it's so important to have you there, having your eyes on the situation, and to have your voice also within your community to talk about the important steps around, first and foremost, preventing the introduction of invasive species, but also to keep an eye on the monitoring and any management that's required with invasive species. So this is a monitoring program using eDNA technology, which we're very excited about continuing to learn more about and deploy as a tool in our overall monitoring programs. But education is a real important part of our ongoing invasive species work, because we need everyone to be part of the solution, and that's keeping these species that are non-native from entering our waterways to the extent we can, and making sure all of our neighbors understand why that is, what the steps they can take, and how important that is. So the whole point of this program is to bring sampling resources to you and get your help in sampling water bodies across the province. We're going to be close to 80 lakes we're going to sample across this ecoregion 5E, and Rebecca will talk a little bit about why this region is of such great interest to us. And this year our focus is going to be on 2 species, which Rebecca is going to talk to you about right now. So I think, here's a bit of the timeline for your interest. We're going to get you kits in the next couple of weeks. So those will be mailed to you. We'll be emailing instructions. Colin's going to do a quick overview, but we'll send the detailed instructions so you have them in hand in the next couple of weeks. But we're going to ask you to sample later in the summer, in the back half of August up to Labor Day, when we think the biggest chances of detection for this plant after a season of growth would be. We'll process the samples in the fall and get you back the results thereafter. So that's the rough timetable. So we're happy to do an early introduction now, and I'll turn it over to Rebecca so she can tell you a little bit about the ISC and about the species that we're looking after. So Rebecca, over to you.
Thanks, Terry. Can you hear me okay? Cool. I'm just gonna have my camera on to say hi, and then I'm gonna turn it off so I don't distract myself. So hi everyone, I'm Rebecca. I work at the Invasive Species Centre. We are a not-for-profit organization that connects stakeholders, knowledge, and technology to prevent the introduction and spread of invasive species that harm Canada's environment, economy, and society. So we've been working with FOCA on the iSample program for 3 years now. This is our 3rd year. In our first and second phase of the program, we focused on invasive mussels like zebra and quagga mussels, as well as spiny water flea. In our first year, we sampled 25 lakes in the Lake Huron, St. Lawrence, and Lake Ontario watersheds with high calcium levels and public access points. We had those specific criteria as we were asking for volunteers. We had volunteers use plankton haul nets to collect water samples, and then we also added this eDNA component as well to our phase 1. Some of those samples were sent back to us, the ones that were taken with those plankton haul nets, and we analyzed them in our lab at the ISC using a sugar separation method, and we tested them for presence or absence of invasive mussels, and then we kind of just used a visual method to look for spiny water flea. The same thing happened in phase 2, except we did 29 lakes and we didn't really have any criteria. We were just interested in anyone in Ontario. So the same thing happened. They used these plankton haul nets, incorporated some eDNA, which we sent to a lab, and then same method. So that was the first and second phase of the program. This year, we're doing things a little bit differently. We're going to be sampling about 78 lakes in Ontario, and we're going to be specifically focusing on eDNA only. And we will be testing these samples for European water chestnut and water soldier this year, instead of mussels and spiny water flea. So why are we focusing on these species? Water soldier to start, it's an invasive aquatic perennial plant that's native to Northwest Asia. It was sold for use as an ornamental plant in water gardens, which is the most likely source of its introduction into the wild. It can spread via human-assisted means, so intentional introduction, so again, in your water gardens, but also by accident through boats and trailering. And then there's natural spread. These turions or baby plants will break away from mature plants and they can actually float downstream and spread that way. So it's actually a prohibited species under Ontario's Invasive Species Act. So it's regulated in Ontario. Currently in Ontario, there are populations within the Trent Severn Waterway, the Black River, which is near Sutton, and in Red Horse Lake north of Gananoque. Most recently, it's been detected in the Bay of Quinte, so this kind of shows the need for broader surveillance throughout select regions of the province. We're generally looking north of Trent Severn Waterway into an east-west line from Espanola to Deep River. And this broad region is water soldier's current distribution. Early detection and rapid response are critical to regional eradication. So your data helps us build our understanding of water soldier distribution, and then monitoring and other interventions where appropriate. So this is kind of how you can tell. It's a submerged aquatic plant, which becomes buoyant during the summer months. As the leaves mature, they become waterlogged and the plant will actually sink below the water surface. The leaves can reach up to about 40 centimeters long. They're sword-shaped, bright green with sharp spines, and form a large rosette or group of leaves arranged in a circle. Flowers, if present, usually not in Ontario, are white with 3 petals. Again, this is really rare in Ontario, so you can't really use this as a main identification feature. The roots can be lightly rooted, but are not always attached to the mud at the bottom. They can be found growing in depths of up to 5 meters, and they can produce those offsets or clones, like I mentioned before. They're very similar in appearance to an aloe plant, a household spider plant, or even the top of a pineapple. So it can be confused with other aquatic plants in Ontario, like native burr reeds, arrowheads, or eelgrass. But none of these have serrated leaf edges, which can be used to easily distinguish them. Next slide. So the issue with these guys is they form dense mats of floating vegetation, which can create stagnant waters and crowd out native vegetation and decrease biodiversity of other plants. They have the potential to alter the surrounding water chemistry, which can harm phytoplankton and other important aquatic organisms. The dense mats can hinder recreational activities like boating, fishing, and swimming. And those sharp serrated edges can actually cut swimmers and the individuals who handle it. So it's a bit of a health and safety concern too. Next, the other species we're testing for is European water chestnut. It's an aquatic perennial plant that grows in shallow or deep water and was brought to North America by a gardener in Massachusetts. It's been found in Ontario and Quebec. And it was early arrival in Ontario with significant efforts by the MNRF to curb its spread. It's also illegal under the Ontario Invasive Species Act, so it's regulated in Ontario. The Ontario distribution includes about half a dozen sites, all under active management, which includes Niagara, Kingston, Perth, and Ottawa. We're also looking generally north of the Trent-Severn Waterway into an east-west line from Espanola to Deep River, because with 2 to 3 populations occurring in the eastern portion of this sampling area, it's possible that it's present in other waterways in this zone. So again, the help and the data that we receive from our volunteers through monitoring via iSample can help us scan a broad area with many suitable waterways for this invasive plant. So this is what it looks like. Has green leaves with sharply toothed edges. It has an appearance unlike any other plant species in Ontario. So the features that can be used to identify it include those floating leaves that are green with sharply toothed edges. They form a densely crowded rosette up to about 30 centimeters in diameter. Their leaf stems up to 15 centimeters long with a spongy swollen section that helps the plant float. Underwater leaves that are feather-like with finely dissected leaf segments. They have small flowers about 8 millimeters long that are white and have 4 petals. They have hard woody seeds, like about 3 to 4 centimeters wide with sharp barb spines. That's that chestnut portion that I guess the name comes from. The viable seeds are greenish brown and sink to the bottom. Older seeds are black, floating, and not viable. And again, these nuts bear no resemblance to the actual water chestnut you'll use in cooking. Next slide. So some of the impacts with water chestnut, similar to water soldier, they form dense mats of floating vegetation, which can crowd out native vegetation and decrease biodiversity. They can also alter the surrounding water chemistry and cause impacts to recreational activities such as boating, angling, and swimming. The hard nuts can accumulate on shore and cause injury when stepped on. They're really sharp, so another health and safety risk with this plant. And with that, I will pass it over to my colleague Colin to talk about eDNA sampling.
Wonderful. Thank you, Becca. Good afternoon, everyone. My name is Colin Casson. I'm the policy manager at the Invasive Species Centre, and happy to be the hand model today as I walk you through what it looks like to do some eDNA sampling. Just a quick bit of background, just a level set for everybody. You know, eDNA, it sounds highly scientific and a little bit intimidating, and I just wanted to explain a little bit on the background of it because it's really super user friendly in this context, and it's, you know, it has limitations. It's not a panacea, but it will really, really help us to continue to evolve our understanding of what is where. And so a little bit of background on eDNA and why we can use it. And this is just a graphic from a fact sheet that's linked at the bottom there if you want to check it out in a little more detail. But, you know, why are we interested in using eDNA or environmental DNA? It has been used for, you know, more than a decade now by research scientists, and really until the last couple of years have we been able to take that idea in a lab context and translate it into a scalable community science application. So we've had great results. We've been doing this for about I guess this will be our 3rd or maybe 4th field season using this tool at some level of scale. And each of the last few years, we've had, you know, really good results actually. Taking people who have no formal training in water sampling, you know, no technical background, no lab coat required, and been able to produce good data. And the exciting thing for us is that the new version of eDNA sampling enables us to send really small kits to people, so the scale opportunity is just magnificent. We've had provincial scale monitoring efforts in in the province of Saskatchewan and in Alberta, and in Ontario as well, and they continue to give us good information, and that's like a very early warning system that can help us. It doesn't tell us with great level of certainty is that species there and how much of that species is there and what do we do next, but it does give us that early warning bell that we need. So if the right conditions are there, then we can deploy another layer of response. So maybe it is a survey team to go out and check out that lake because you've got a positive hit on water soldier, God forbid. But a little bit of background on why to use eDNA. It's easy to use, it's scalable, it's something we can ship, like I say, kits across the country and be able to collect good data. So from my end of the spectrum, it also helps us be very cost effective and efficient too, and engage community science and people who have a real interest in one particular geography like yourselves. And really this is just scalable. It's a good way to kind of democratize science in a sense and bring it to people who have an active interest in wanting to figure out how can I contribute. This is a really easy opportunity with a quick webinar today and about 8 minutes of work from your local dock or boat launch. You can help us understand what's going on in your lake. We can give you that data, and we can also, if the conditions are right, report that finding to the right authorities to be able to support that management decision. So that's the quick and dirty version of eDNA. We can collect eDNA from air if we're collecting pollen. We can collect it from water samples, which is of course what we're going to be doing today. We can collect it from soil. We can collect it from snow. All kinds of different sources, processed and used in different ways to basically answer different kinds of questions. But for the sake of today's conversation, as Terry kicked off, we're interested in aquatic invasive species, and as Becca noted, water soldier and European water chestnut are our two high priority species right now because we really need to better understand their distribution in your part of the province. Thank you. So what can it do, what can't it do? Again, it's not a perfect tool, but it is a really useful tool if used correctly. And if we can understand how it can be used and where its limitations are, it helps us all get a little bit more out of this process. So basically some things it can't do. It can't tell us if that species, maybe it came from a fish, that eDNA that we picked up in that sample that you collected. It doesn't tell us if that fish was alive or dead. Doesn't tell us if there was one fish or many fish that are in a reproducing population in that watershed or in that water body or not. Doesn't tell us if it was a juvenile, doesn't tell us if it was an adult. Doesn't tell us if that species is resident or established in that water body, or if just it was passing on through en route to another lake. And it also has a bit of a difficult time in distinguishing between hybrids of species. That's a finer point, only applicable for certain species. So those are some of the limitations of it, but what it can do is a positive detection can tell us that there was eDNA or environmental DNA of that species we're interested in that water at that time. And that's enough to really start to inform how we deploy some resources at a provincial or national level in terms of the follow-ups, because we can't be everywhere, we can't survey every lake, of course. But if we can use this as an early tool to triage how we're using our resources and following up on those lakes or those communities or those regions that give us a positive hit and maybe are require us to have another look, that's how we're trying to use this tool. And a negative detection doesn't tell us that it wasn't found it's not in the lake. So I just want to be very clear that if the sample that you have comes back negative, it might be a function of the species was there and our sample size was insufficient to pick it up, or a variety of other factors. Maybe there was a large rain and that system flushed through. Of course, the solution to pollution is dilution. Same holds true for eDNA that's floating about and being shed by any living or dead organism. That might be a condition too, where it's a little bit harder to pick it up. And also we might be sampling one part of the lake when the species is present and established in the other part of the lake. So, in terms of a good question we always get is how sensitive is this thing? Can it, one drop of water tell us everything that's happening in the lake? It really depends on the species we're sampling for and the right conditions. Was it raining? Was it not? Is this part of the spring freshet? What's going on? But a general reasonable rule of thumb is maybe about 500 meters or so is roughly the sensitivity. So if you're within, let's call it a half a kilometer, very approximate guidelines, but it's a useful concept. If we're within about a half a click of that species being present, there's a reasonable chance that we'll pick it up in the sample. If we're 10 kilometers away, it's very unlikely. Again, that's just one organism spewing eDNA into the water. And again, the solution to pollution is dilution in a sense. So it's a hard time for us to pick it up well downriver. So just a little bit of context there on what it can do, what it can't do. Again, it's a really useful tool. It does not answer the question with the highest level of certainty, is that species present or not? But it's a really cost-effective tool for us to be able to do a broad-scale monitoring effort, which is really helpful. It's a first line of defense. Next slide, please, Michelle. Just a few notes here, I guess, before you hit the water. Your package, as Terry mentioned, is going to have detailed information similar to what you're seeing here today, but I've just stole this out of last year's sampling guide. So it's very similar stepwise, but it's not going to be precise, and there will be some finer points here. I think last year, or sorry, with this other project that we used this guidance from, I think we were collecting from 3 different parts of the lake. So there's some finer points that are going to be modified. So the stuff you're seeing on screen is not exactly what you're going to see in your instruction package. So just want to make note of that, just so there's no confusion here. So appreciate you taking notes today, or if your memory is so good that you're remembering that thing on the screen said collect 3 samples, it's going to be different in your sampling package. I just wanted to show you, for the sake of being familiar with the content, there's a small section in that application guide that just gives you a few tips. Some people are more familiar with working on water or near water than others, so just want to make sure everybody's safe and happy. So, some things to consider before you go. I mean, first of all, if it's a beautiful day and this is a recreation opportunity for you, take all the time you'd like to out in the field, but would recommend you giving it a quick read, the protocol. And we've got a sampling video that can help you just refresh yourself with the steps. What I would recommend is kind of having it up on your phone so you can follow stepwise one at a time. It's numbered, and I'll show you what the step-by-step instructions look like in a moment. But just giving it a quick read before you hit the field will help make sure that you're being efficient and having a good time out there. Obviously, simple things like check the weather, just make sure your lake conditions are safe. For the type of sampling we're asking proponents to do, or you guys to do here today, we would ask you to go, we're trying to skew this to finding these species. So where do we know aquatic invasive species are most likely to first show up? On your public boat launches for one. So that would be a great kind of a place to go out and sample. So, no need to get the boat all geared up and head over to the other side or the center of the lake. I think we're looking to try and skew this towards where we think these things would first show up in a most likely scenario. And because many of your lakes would be landlocked and not part of large flowing systems, I would presume, I think the best public access point, or the best opportunity for us to find these things, would likely be close to the most popularly used public access points. Public boat launches are really the best example. If you don't have a public access point, that's okay. Just choose, hopefully that insulates you from the probability of new aquatic invasive species being found in your lake. Let's cross our fingers. Hopefully you won't find any. But where would you sample in lieu of that kind of a location? I would think to go look at, the right kind of habitat in that scenario. So we're looking for kind of weedy bays. Again, maybe, areas that are hot spots for anglers. Those would be the kinds of conditions that we might find water chestnut or water soldier. So as long as you can be within roughly 500 meters or so of that kind of a situation, sounds like a good spot to sample to me. But again, look for those public boat launches or those kinds of places we would expect first aquatic invasive species to pop up. That sounds like it's your best spot. And yeah, I mean, obviously, just like near water safety is pretty important here. So let's just be aware of that. I think when we were developing this specific text, again, we were asking people to go hit the public boat launch and then go do a deep water sample, because that priority species for this use was for fish. But we're looking for aquatic plants, so let's go to where we know they would be. And again, we're doing roughly on water sampling. You might wade into shin deep water if you choose, or maybe it's a public boat launch with a dock that you can go and just kind of be at the side of the dock and scoop up some water to sample from there. So those are great examples. If you do want to take this into water and make a bit of a day trip out of it, by all means, take the boat out. But of course, just be safe when you're working on water. It's my only request, please. Next slide, please, Michelle. Okay, so this is what you're going to get. So Terry's going to ship you a Ziploc baggie full of things like this. Just a good old fashioned freezer baggie with a bunch of equipment in it. And what's in it is what you're seeing on screen here. So I've got it all laid out in front of my desk. I'm going to give you the best hand modeling I can. So I'm going to show you kind of one at a time what these look like, but there's some pictures in your guide of what each thing is called and looks like. So hopefully there's a lot of familiarity there. But a quick rundown. So you're going to get some gloves. We're processing these samples for our aquatic invasive species of interest, not for human DNA. Obviously, eDNA is just so sensitive that even if we're following a really strict procedure and protocol, if we're new to it, we're going to pick up human DNA. No, the tests we're running these samples for are just looking, it's essentially a yes or no question. Was water soldier present or not? Was water chestnut present or not? So don't worry too much is my guidance on contamination. Please, do your best just to keep the sample as clean as possible. Try not to drop it on the bottom of the boat, little things like that. But the gloves are just kind of, they're just, mostly just to make sure that we're doing our best. Understanding perfection is not going to happen here on keeping the sample free of any external contaminants. So gloves are included in the package. I think we do our best to try a medium, hoping that it makes everyone equally as comfortable-ish. I know there's some small hands and some extra large hands too. So we just ask you to do your best. I think we have either medium or large that tend to go in most. So if they're a little bit big and baggy on your hands, I apologize in advance. Hard to customize it. So that's the gloves. You're going to get a syringe, which looks just like this. It is just a plastic tube, and it's 60 mils or so. And if you're strong enough, you can pull it open, squeeze it closed. On the tip here, it's got a lure lock fitting, so it's kind of a screw-in end. It's not a sharp. It's plastic. There's nothing sharp here or anything like that. So no, sharp issues or opportunities to get a poke. It's just plastic and it's a dull point. Next, you're gonna have a filter capsule. So that comes in a baggie like this, which I've just ripped open there on one side so you can see what's going on. And if I had gloves on, I'm doing this in a non-contaminatable way. Sorry for the glare on the background, but basically it's just a little filter cartridge. And it's a plastic housing that's going to screw onto that. And I'll show you in a minute. It's going to screw onto your syringe and you're going to squeeze water through it. And inside this plastic filter cartridge here is just a little piece of filter paper with a known pore size. So it's just a piece of filter paper that has a tiny pore in it. And it's going to take maybe very turbid or very colored water that has lots of sediment mixed up into it, and it's going to suck, it's going to hang on to all those sediments or all that eDNA or anything else that might be floating around in that water, algae, you name it. And what's going to come out of that filter is just water. So we're going to leave the water, but keep the DNA or keep anything that's found within that water. So that's the point of the filter here. In kind of more research applications, so not the version of eDNA sampling we're doing. People have had pumps and filters that they need to, clean between samples, and it's been a real challenge. But this is ultimately the reason why we're able to do this is because it's a very low tech and ultimately shippable version of all that equipment of yesteryear that used to be relevant, and now we're able to just again filter water using just this small micron filter. That's the filter. A large tube here of phosphate buffered saline. So this is just a tube with a clear liquid. It looks like water. It's called phosphate buffered saline. We include, as we should, the MSDS sheets, but really phosphate buffered saline is effectively salt water, but it's of a known pH. So that's why we pay a little more for it than we would some distilled water. It's just ultimately this is a way of rinsing out once we've filtered out a bunch of lake water there. This is our way of pushing some known pH water through, and basically just our way of cleaning out that, flushing through that filter just to make sure it's nice and clean as we prepare it and store it before the lab can get it. So this is the large tube. It's called PBS, or phosphate buffered saline. Again, effectively salt water. We are asking everyone to wear gloves, but just in terms of the human health considerations and safety considerations, good to speak to. Of course, this is a very safe product because, again, it's effectively salt water of a known pH. So if you get a little bit on your skin, no concerns. There's the MSDS sheets, which we provide, and I think a quick flush of water, but nothing toxic or hazardous in that regard here. It's a pretty, useful product and user safe and friendly. So phosphate buffered saline. There's 2 tubes that we provide you with. There's one here that's the big tube and one here that's the small tube. So, one has a 1 mil capacity and one has a 60 or 50 mil capacity. So when we say big tube, small tube, that's what we mean. Okay. So small tube is the next one. That's the preservative. So we have a couple different colors of tubes. No concern there. But this is just what's after we use that phosphate buffered saline, the big tube liquid. We're gonna add this, and basically this just kind of stops any decay from happening. Again, there's MSDS sheets just to make sure everybody's aware of what's in the kit, because it's a good question to ask. Obviously, if somebody sends you a liquid of unknown origin, so. Have a look there, but basically what this does in terms of our function here is just it acts as a preservative to the sample to make sure that it doesn't degrade too quickly. So so this is our preservative or buffer is what we call it. So we try and be consistent in our language, but big tube, small tube, it's hard to mix them up. The liquid from the small tube, you're going to get out with a pipette. And they're just small disposable plastic pipettes. So it's just a little bulb on the end. And basically, I'll show you, kind of give you the step-by-step in a moment, but that's what a pipette is, as obviously you know. And there's 2 caps. So we call them cap A and cap B. There's pictures there which distinguish them, which is going to be better than anything I can hold my hands up to the camera to show you. But 2 different versions of the caps. And it's pretty user-friendly because each of these caps can only go on one end. So the ends of the filter, these caps are for is to lock in the filter on either end. And the good news is they only each fit on one end, so there's no way to mix them up. So it's pretty user-friendly in that way too. So there's 2 different caps, cap A and cap B, if you like. Call them whatever you want though. A couple small baggies, plastic film. Sorry, that's a typo. We're not sending any plastic film, so not to worry. There's 2 baggies, and we call them Whirl Pack bags. They're just, I would guess it's like 500 mils, roughly something like that, half a liter, a quantity, something like that. And there's 2 baggies. And the reason why we give you 2 baggies, I'll touch on in a second, but one is your sampling water baggie. So go ahead and take that once you get it to the field. You just tear the top off because we know it's going to be sealed inside. And open up the bag, scoop up some water, and away you go. So more on that in just a second. And the second bag, it's the exact same, it's the same kind of bag, but this is going to be what you're going to send us your filter. So the end product you're giving to us is just a baggie and your filter cap, which hopefully has a whole bunch of eDNA in it. We'll send you a pencil, a data sheet, and a return envelope too, so that you can send us back in that plastic baggie. Just throw your filter, which is covered in a plastic baggie, and mail it back to us in a pre-addressed stamped envelope. That's what you're getting from us. Now let me show you how to use it in a second here. So this is the sampling procedure that you're going to receive a copy of. This is, again, the old version from last year. So give me a little bit of time, if you don't mind, just to do a couple quick tweaks and updates to make sure it's exactly telling you what we're looking for you to do. If you see 22 steps, my gosh, I didn't sign up for that. Just want you to know it is, we didn't write like, make sure you take a breath and breathe. But we do kind of over articulate them. Hopefully that doesn't scare anybody off, but basically in terms of how long is this going to take you, best guess I can give you is, 10 minutes, 11 or 12 minutes if you're enjoying them at the moment. It's not a long sampling exercise by any means. It's really for me just about having people who can get to the site, because once you're at the site, no problem. It's a quick jaunt and a quick sample collection. So we do go step by step, but I'm going to give you the show and tell version of this in just a moment here. So hang tight, but just wanted to show you this because it's going to look visually very similar to what you were seeing. Next slide, please. And we've got a little video. It's me as the guest appearance in the video. So I'm sorry, you're going to see a lot of Colin in the next few minutes, but we'll give you a quick version of that. And then I'll take you through the step-by-step in a moment. So if you could hit play, Michelle.
Hi, thanks for your interest in helping us monitor for aquatic invasive species using a cool new tool called environmental DNA. The technology has come a long way in a short period of time, and it's enabled us to use groups like you, citizen scientists and community science, to go out and monitor lakes and water bodies like this for priority aquatic invasive plants and other species that are of concern to us. We take that data and it helps us better understand where species are, or hopefully where they're not, and ultimately make management decisions based on that information. So to participate in this program, we've just shipped you a kit. Within each kit, you're going to have a smaller envelope which contains a bunch of information for you to go out twice and help us sample for aquatic invasive species. You're going to use things like syringes, tubes, rubber gloves, pipettes, and a bunch of other stuff which we'll cover in a few minutes to go out, sample your water, and ultimately we're going to get some information on what species are in your lake. Thanks for helping.
Okay, so I'll let this run for a minute, but just for the director's cut. So we're out here sampling. This is the Black River, but you know, your lake would be your lake, of course. And again, if you're interested in sampling from a public dock, maybe a public boat launch, that's really the sweet spot, I think, where we're most likely to find these species. So that's what I would encourage you to try to do. And essentially, what I'm doing here is just collecting, again, one of those bagfuls of water from our lake. And perfect, if you want to stop it there. Awesome, thanks, Michelle. And so why don't I walk you through kind of what that looks like. So I'm going to give you a quick demonstration step by step. Again, you're going to see this, you're going to get the written instructions and you're going to have this package. It's a little bit easier to do once these things are in front of you. So hold with me if you don't mind for a couple of minutes here. I just wanted to give you, a first introduction to what does that look like. So I'm at the public boat launch. Here we are, forget about the background. And I've got my big Ziploc bag that I brought with me of what FOCA has shipped over to you. And I've had a quick look through the kit before I get out there. I've given a quick read and a refresher through that document, which has the 22, hopefully less steps that just articulate, one step at a time. What are you doing and why are you doing it? And I'm at the boat launch and I'm ready to go. So what do? Well, first, the step one is going to be grab your baggie. And I showed it before. I'm sorry I did it, but I just ripped off the top. These bags are just sealed because we want to make sure when they ship them from the manufacturer that we know there's been nothing in them. So first use, we're going to tear that off. And this just becomes our water collection bag. Again, there's two of these bags, two different purposes. One is you're going to scoop some water, and the other one is what you're going to use to protect your filter capsule at the end of the day and ship it back up to us. So we've just scooped up a bunch of water and oh my gosh, through the magic of television, if you would imagine that this glass of Gatorade is the water from our lakes. This is what we're going to be sampling. Please, I don't want to soak my floor on my desk here. So just imagine this is within this baggie, if you would. So we've got our sampling bag full of water, and it's blue water. So what are we going to do next? Okay, wonderful. So it's time to glove up. So like I've got a feature in cooking shows here. See how this works. So again, it's like 8 minutes in the field. We'll do the quick version. So hang tight for another couple. So I'm gloved up. And again, this is really just about doing the best we can to prevent any contaminants from entering into the filter. Again, perfection is not our goal. Really, we're just going to process these samples for two things: water chestnut, water soldier. Are they there? Yes or no? So, don't worry about it if there's a little bit of a contamination issue. It happens and it doesn't impact the quality of data we get for the species we're interested in. So what are we going to do? So we've just opened up grabbed our filter package here. We've opened the baggie and out pops tool number one. This is our syringe. So again, this is a 60 ml syringe. We're just able to suck up a bunch of water. And once we get the filter cap on there, squeeze it through. So I'm going to fill this up. Again, let's pretend it was from the bag of water, but it's actually a mason jar. Prevent spillage. And all I did there, if you're familiar with children's Tylenol, you're just filling up the syringe here full of our lake water in this instance. And what am I going to do now? Well, I want to filter it. I want to know what's in this water. So I've grabbed my filter here. And again, there's two different ends on this. It's only one end that'll screw on here effectively, so you'll know. And these just twist in. It's just a lure lock, so it just twists in. And what am I going to do? Well, I've got a disposal cup here, so that's where that's going. But all you're going to do is squeeze the water through the filter. I don't know if you can pick that up. Anyways, so I'm just filtering through that Gatorade here. Turns out Gatorade is harder to filter than lake water. This is when I realized I should have done this with Gatorade in my practice run. Okay, so we've just done one run through here, and let's pretend that I was able to get the full syringe through here. And if you'll notice, I don't know, it won't pick it up, I'm sure, but there's a bit of a light blue tinge to this already. It went from a white filter cartridge to like a light blue now with lots of sugar crystals in there. But that would be, you know, the sugar crystals are your eDNA from your lake. So that's great. So that's all we want. We don't want the lake water. All we want here is just our eDNA. So we've done that once. The thing is that that's only 50 mils worth of sample. And I'm trying to get as much information from your lake as possible. And in the event that water soldier or water chestnut are in your water body, I want to, you know, give me as much data as you can, please. And data in this case is volume of water sampled. So the more samples we can take, if we replicated this exercise 100 times through your lake, or if we just, you know, filtered 100 squeezes of this tube through our filter, either way, it's giving us more data. So that's what I'm asking you to do, is filter as many replications of this as you're able to do comfortably. Now, you know, water bodies are so different, as you know. Like a tea-stained lake, you might only get maybe 2 or 3 of these through. Gatorade, I got about a half a squeeze through. But if you're, you know, in a beautiful, pristine, clear lake, you might be able to squeeze, you know, 5, 6, 7, 8, 10 squeezes of this through the filter before the filter starts to clog. So I can't give you great guidance on how many times your lake is going to be able to squeeze this through. I would love it if you could at least do one. Hopefully you're not filtering Gatorade like me, but just do your best is all I would guide you on in terms of how much water can you filter through this cartridge. But again, let's assume that it was a clear lake, not Gatorade lake here. And what I would do is just fill up another syringe. Okay, so just filled up a fresh 60 and just do the same thing. So I've unscrewed that cap, so I'm going to screw that cap back on. Again, it only locks in one way. And again, I'm just going to squeeze this through. If I was strong enough, I would anyways. And that water is then going to go through, or the Gatorade in this case, is going to go through this filter again. And again, fresh water or more or less pure water would be coming down the other end. I'm just capturing in this filter everything I'm curious about, eDNA of all species. Invasive species like the 2 we're going to test for, native species like the, you know, the great diverse fish communities hopefully that you have, or native aquatic plants. You know, in theory, if the lab was doing different tests on the same sample, we should be able to pick up anything that's in that water body. Or even eDNA that's washed off the shore and into that water body too. It's a really interesting, remarkable, and sensitive tool. But again, we're interested in the aquatic invasive species in that lake, so that's what we're processing for. So as I say before, you know, we've been processing as much of a sample size as we can. So let's say you were able to do, you know, 4, 5 squeezes of this filter, of this syringe of water through this filter. That's a wonderful sample. So thank you for your efforts in that regard. And once we get to the end of that, what are we going to do next? Well, we've got lots of now of eDNA in here. What do we need to do? We need to prepare it for its journey home. So so this is empty, as you see, and in the other hand here, I'm holding my filter cartridge. What I'm going to do, while they're still disconnected, I'm going to suck up a whole bunch of air instead of Gatorade or water this time. I'm going to screw it on, so my syringe is now loaded with nothing but air. And I need to purge any remaining lake water through the sample. So I'm going to grab it on here and I'm just going to push air through here. And it might take a second here, but you're going to squeeze out just the last few drops of lake water that's in there. And again, it's capturing all that eDNA, whatever's left in there in the filter cartridge. So that's wonderful. And I'm just looking at this now. I got about 90% of it. So let's do another load of air real quick. And we can get you on your way here. Okay. Wonderful, wonderful. Okay, I've got all that Gatorade or lake water now flushed through here. This is an empty cartridge. It's more or less just air, but within that filter paper is still all that eDNA. Any water that trickled through was leaving its eDNA from its fish, zebra mussels, whatever were in there, and now they're captured in here. But again, this is about prepping this guy for sample or for the road. So this is a step that, again, it's kind of a finer point step by step, but it's articulated in the instructions. What are we going to do? This is a bit of a hand dance here. So I'm going to take the syringe top out. I'll show you why I do this, the method to the madness in a second. And I'm going to screw on that filter onto the bottom. All of a sudden, that filter is acting as a bit of a plug. So when I add that new phosphate buffered saline, that big tube, all of a sudden, because the filter cap is on here, it's going to stop it from running down onto my floor. So that's why I'm doing this. But again, it says step by step why you do that in the methods. So I've unscrewed now. Sorry, it's a bit of a dance here. You'll get the hang of it, I promise. I've unscrewed the big top here from our phosphate buffered saline, and I'm going to add it to this. And again, the function of this is just, it's a known pH. It just helps us make sure that there's, if you have acidic water or basic water or whatever you've got, it just helps us make sure that this sample is kind of uniform and processed in a consistent way between all samples. So again, this filter cap at the bottom here, it just acts as a plug because I just added this water from the top here, this phosphate buffered saline. Now I'm going to put the cap back on because I need to filter all that through. So I'm going to really regret that. And cut to an infomercial. This is when we would do that. And I'm going to use the magic of television and totally cheat. Let's pretend that I just pushed that through. I'm sorry, my arms are wimpy today. So we've just flushed that through, and now what is through here? Nothing but that phosphate buffered saline. So again, it's just that known pH. And we're going to do the same trick we did before so that we can flush this again, empty with water. So once again, the filter is off. I'm going to load up some more air. Probably seems very complicated the first time you see it. I get it. It will make sense when you've got this 22 steps in front of you. So this is full of phosphate buffered saline now and the DNA in the little paper cartridge that's inside. We've loaded this syringe now up with air. What are we going to do? Just squeeze it out. So do your best here. Much easier to push air through this than it is liquid. Okay, sorry, I'm trying to do my best with the camera here. Okay, and so now this has been washed, if you like, with phosphate buffered saline. So we know it's not acidic, it's not basic, it's the known pH. That's the function of that. That's why we do it. And okay, so now what are we going to do? All we need to do is add that small tube now, and we're right at the finish line, I promise. So what we're going to do here is, if I just added that liquid now, of course it would just run through. Again, kind of similar to what we had before. We had a bit of a vacuum lock on this. So what do we need to do? We need to add our cap. So down to the bottom here, all we're going to do is add that cap on the bottom, and I'll do my best not to block the camera. I apologize if I do, but basically I've just added that cap down to the bottom, and away we go. So now we can add our small tube of liquid, that preservative liquid, to this. So hang tight, if you would, for 2 seconds here. You know, there's a reason why we have children in this world, and this would be probably it, because a second set of hands isn't the worst thing. So if you have a sampling buddy, child, grandchild, this is it. Great opportunity to engage them in some lake stewardship. So what we're going to do here is, I've got the small tube, which I showed you before. Again, it's the 1 mil versus the 50 mil. It's David versus Goliath. So here's the David tube. And I've got a syringe. So I'm going to dip the syringe into that liquid, and I'm going to suck it up into that syringe. And again, this is, I got about half of it or so in that syringe now. Sorry, pipette, I'm saying syringe. And what I'm going to do, oh boy, grab that filter cap, which again has all of that eDNA in it. And then we washed it through with that big liquid, the phosphate buffered saline, the salt water. And now we're just going to add this preservative so that the lab is happy when they get a really good quality sample. So what I've done is I've just taken the tip and pushed it down into the filter cartridge, and I'm going to squeeze it out. So all that liquid is draining down into there. I'm battling some bubbles here, so I'm going to go nice and slow. And again, all I'm doing here is just making sure that this is safe and happy in its mail trip back to the lab. So off camera here, I've just taken my pipette, I put it back into the small tube of preservative here, and that small tube is now empty, that 1 mil is now empty, and I'm just going to repeat. So keep adding it to that filter. We're going to send it home to the lab in a bath full of this preservative liquid. And again, that's, you know, just an extra step here to make sure that when it gets to the lab, that DNA hasn't degraded. Because by nature, eDNA is, the second it's off the living organism, its clock is ticking. It's not going to hang around in that water body forever and ever. You know, so it depends on the conditions. So I can't even give you a good estimate as to how long it would last. But we're talking, you know, months or maybe years, but not decades. So the quicker we can get this sample to the lab, the better. And by adding the preservative, it just helps make sure that the status quo is maintained so that we can get good data out of the sample. Hang tight, we're almost done. So again, just to recap what we did, we just added that small tube of a preservative to this filter. It's now roughly full, like, 2/3 of the way up the cap of the filter here, full of liquid. It doesn't need to be full to the top, don't worry about that. And all we need to do is cap this off now. So there's the other cap. Again, this is the only, each cap only fits on one end, so no worries about, I don't know which side, it'll lock in for you, no problem. So I've just screwed it on here, and now I've got that new cap on this end. I've got my original cap on the bottom end here, and this is a great sample. You just collected an awesome sample from your water body, so thank you for doing that. What are you gonna do now? Get your grandchild or your child or your friend to pass your other bag. This is the unopened bag. And what are we going to do? We're going to rip the top off. They will maybe. And we're just going to put this filter into the bag before we can put it away into the mail kit. So I'm going to drop that in. And I was smart enough to pre-label, and many times I'm not. So tip that I'll include in the written instructions for you is a good idea to label your bag before you get out there. And we're gonna roll down, and use our tie here to kind of do one of those. So there we go. So this is our sample. This is what we're gonna put into a envelope, a bubbled envelope here, and it's gonna go back to FOCA, and it's gonna get to the lab shortly thereafter. And then our lab techs are gonna take this and run a PCR test on it. Same kind of PCR as, we're all unfortunately familiar with by the end of COVID, or this stage in COVID. So all you're gonna do here is, again, put this into your envelope. Let's address Terry personally. Relabeled, pre-stamped, so it's nice and easy for you. Way to go. So this is what we're going to get back from you. Again, once it arrives in the lab, we're going to be able to take that filter, crack open that plastic, take the under controlled conditions, take out that white filter paper, and ask the question through a series of lab tests. Was that species there? Yes or no? Was water soldier, as Becca mentioned before, there? Yes or no? Water chestnut? Yes or no? And so we should have a reasonably confident answer on where you sampled, at least. And hundreds of meters on either side, 500 meters, let's say, to the left and to the right. Was water soldier present, yes or no, at that time that you collected the sample? Again, in terms of interpreting those results, just want to be clear in terms of what a positive or a negative sample means, and we're happy to give you guidance, of course. And if you got a positive sample, you're going to hear from us. Not to worry, but I will worry. But yeah, so what that positive sample means is that there was eDNA present at that time. Good question. I see a couple questions coming in, so I'll try and do a quick one here. But so can that syringe be used again? Again, I mentioned earlier, the older version of this technology required a whole bunch of reusable equipment. We've designed this in mind so that it's a small baggie. I know there's a bit of plastic waste in here, which I see in here. It's a small baggie. All of that is disposable. The only thing we would like back from you is, again, just mail me, please, if you would, that envelope which has that eDNA filter in it. This is all we need back. So please feel free to dispose of the rest. It's not really useful material otherwise. So yeah, and we really, really appreciate the work that you put into collecting this sample. Again, it doesn't answer with every level of certainty, is there that species present in your lake, yes or no? But it's, again, a really effective tool for us to do a broad scale monitor effort across a particular region of the province that's notable for us, with those two species in mind, which, again, as Becca noted, are provincially regulated species and really do require a hefty response if and when we find them. So, I would hope certainly that we're not going to find a whole bunch of positive lakes. I'm sure Becca and Terry are on the same page there. But we don't know until we get that data, and you're helping us get that data. So we appreciate that effort. Yeah, I think that's all I've got right here.
Great, thanks, Colin. Good hand modeling.
Try my best.
I had no idea. A couple questions for you. Can't we just sample for everything and anything since you've got the sample already? And I guess our answer is that really we're funded just to do the two. So just, I guess that was I've already answered that one, but
Yeah, and in theory, yes, but again, it's about deploying fixed resources into the right too. So that's why we've done it. But in theory, if the question is theoretical, that sample could tell us, you know, is species A, B, or C there? Native, invasive, doesn't matter.
Sure. So another question was, do you have to take the water all from one spot, or could you hedge your bets and take the water from several different locations?
If you can get, I would say it's best to collect from one spot. I think if you could try and skew it, we're looking for where are we going to find that thing. So a public boat launch or like the most likely condition. Now, if your lake is such that you've got a public boat launch and it goes deep and the shores are really, really, you know, really deep at that part of the lake, there's not a whole lot of plant growth zone. You know, maybe you say, forget it about my direction there on that public boat launch, you know, would like to standardize it as best we can, but you know your lake better than I do. So if there's a weedy part of the lake that's more appropriate, and again, that boat launch is ruled out because we're looking to find as close to these plants where they would be as possible. So, you know, I'd leave that to you, but I would strongly encourage in the normal sense, most lakes, I would hope would be the most, you know, high public access launch points. That's where we're going to find these things first.
Sure. Why the bag? Can't we just take the syringe of water out of the lake?
You can. I just find it's a bit difficult. It depends on your kind of setup. You know, if you're on a dock and the dock is a bit raised, it's usually just a bit easier to have person 2, which is really helpful to have. Scoop a bag of water up and just be available to support you in that way, as opposed to the reach down and reach in. And again, it just kind of helps us standardize the process and be a bit more clean with it. So I've found it useful, but we're really just interested in the amounts of water. So to each their own, but you know, we found this to work pretty well by doing this in the past.
Okay. Okay. We've got some folks that are actually doing their own eDNA testing and using nature metrics who don't have aquatic plants database. So they're curious how we're doing it because these other folks, a private lab does not.
That's correct. Yeah. So there's a few. That's the most widely used and I think biggest eDNA service provider lab-wise, private lab, I should say. There are, so you know what that test is, that species present, yes or no. It's called an assay, ASSAY, and the different labs have access to different assays, and some private labs, when asked in the past, the response I've got is we just don't, either maybe we don't have that assay or we need to develop that own assay, because ultimately if they tell you, if they process your sample with that assay, they need to be able to vouch for it. They need to have done the background check on it. It's a validation thing. So the lab we use, which is a government affiliate, enables us to be able to test other assays that researchers have developed, which again are publicly available, and private labs are able to set their own list of species they process for and not. And I wish more would be able to test for other species, but if we're interested in plants, which we are, it kind of limits and restricts which labs we can use. And the lab we use is a government affiliated lab, which is why we're able to deliver this service at no cost.
Great. Should we keep, or could we keep the syringe and reuse it, or would we just say don't do that and wait for next year if we do another program?
So. Yeah, and you know, put the screws to me to be able to find a way to find more sample size and collect samples from more parts of your lakes. That's where we'd love to take this. If we can demonstrate success year over year, I'd love to see scale. So I appreciate that. But really, the thing is the disposable filter piece. So I don't think you get very far. It's, you know, to be quite transparent, I think it's about $31 a kit in terms of the equipment, and the filter cartridge here, I think it's like 19. So everything else is pretty cheap and disposable. You know, downsides of the plastic waste, but it's not worth it to keep it. And also, there's again that contamination issue between piece A and piece B. So, yeah. Great.
Large lake, multiple samples, better idea, or I guess your advice has been take your best shot at where you think that the plant might be present. That's
it. In a perfect world, unlimited resources here. We'd love to be able to boost sample size within a lake, and it's something we can take away as feedback for next year when we're trying to figure out how to design this. So I would encourage you to make note of that maybe. But yeah, I would say, you know, take your best shot at the one spot you think it's going to pop up. If you have multiple public access launches, choose the busiest one, choose the one that, you know, there's the lineup at the tournament of people maybe coming from pretty far away. I think that's the right approach. You know, we want to be as we want to skew this to finding these things. It's not about a random sample at your lake scale. It's really about knowing what you know about how aquatic invasive species move around, which is watercraft movement primarily, and people who move to many different lakes in a short period of time before they come to your lake. That's where it's most likely going to be found. So high flow public access points is your best bet.
Sure. There's a few questions about the multiple samples. Just to be clear, there's going to be one sample taken in this year's program, so just a single sample from your best spot.
That's right. I'm sorry, the video did say 2, which I realized after it said it. So just to be totally clear, exactly. One sample is what we've got the opportunity on. Would love to take that feedback back for a future iteration of the program, but one sample, one lake.
Okay. Dam outflow might be a good choice. If there's a single outflow from a lake. That's a question. Is that a good spot?
I think it's reasonable. I'm going to assume that there's no one public access launch, or maybe it's just launch off the end of your property or right of way, something like that. So if that's the condition of your lake, it's relatively insulated, if you like, from invasive species introductions. That sounds like a good spot, but I would skew towards those public access points first. So think public boat launch, think municipal launch. Maybe it's a private launch at a marina and that's the only access point to your lake. That would be another good spot too. But again, you're looking for the highest traffic boat launches with the most people from the farthest away.
Okay. Again, single sample per lake, so that we're not doing multiple samples on any lakes that I know of. When I look back at the list, we're not doing multiples on any of the lakes.
Yeah, that's correct.
A couple questions just about handling, because we want to preserve the sample, as you said. So what are we labeling on the bag? You mentioned labeling the bag.
We'll give you written instructions on what to write on the bag in that. So that'll be part of the before you go portion of the instructions. But just simple things like, what's your lake name? Maybe we can give you a sample collector name just so we have a bit of a uniform thing. A date, but again, we'll give you all that information in advance, so it'll be in your information package. So you just need to know the name of your lake, the date you collected the sample on, and away you go.
Great. And as far as handling it, when it gets to FOCA, we'll be refrigerating it. But in terms of the person collecting it, what do you recommend they do between, like, what's the timing between collecting it and mailing it? And should they be refrigerating it in the interim?
As best you can. I think every little action helps. Critically, if you collected the sample on a Friday and it stayed out for 2 days and then you mailed it on a Monday, it's not the end of the world, but it would, we're trying to collect the best data we can here, so let's go the extra mile. So a good idea would be to take that tear envelope addressed to FOCA here with our sample cartridge inside and just put it in the fridge. If you can, just hold it cold. That would be ideal before you get it to the mail. So a good helpful tip too, which is just a finer point we'll try and mention in the package as well, is ideally don't ship it on a Friday, because of course that's going to just sit in a hot metal box for the weekend, and then maybe it'll be picked up on Monday or maybe not. So would encourage you, if you're there for the weekend, you're collecting your sample on a weekend, just keep it in the fridge until Sunday afternoon, or Monday even better. Bring it home with you and get it in the mailbox as of Monday. That would be preferable, because then it finds FOCA by the Friday, ideally, and then finds their fridge too. So again, it's not the end of the world. The sample is still good and robust enough because of that preservative we add, but let's take every step we can to get good data out of this if we're going to go to the trouble of collecting the sample. So collect the sample sometime over the weekend if that's your approach, and get it ready to mail Monday morning. That would be amazing.
Great. And we're a little over time, but there was a question about the water depth. It doesn't really matter in terms of capturing the eDNA, I don't think. It can be deep or shallow, but we think, again, we want to be in a shallow area, probably is where where the plants are likely to be growing or present.
Exactly. Yeah, I think 4 feet, 3 feet, 5 feet, something around that depth range. That's where we find both of these plants. So let's go looking for them there.
Sure. Okay, well, I appreciate that, Colin. And again, the hand modeling. there will be other questions, I'm sure, and you can be sure that we will be in touch with the detailed instructions. The up-to-date current 2023 instructions will be in your inbox in the next couple of weeks, along with your kit, which will come via the mail. And if you have any questions in the meantime, you can contact programs at foca.on.ca. And yeah, if you have any questions anytime of what we're doing around invasive species, this program or otherwise, or any other questions related to our programs or what our work with ISC and others, please just contact the office. I'd like to thank all of you for joining us today. It's again absolutely irreplaceable value that we have when we've got volunteers that are all over the landscape that are willing to take on this challenge and help us with this important work, and important in our own communities for our own sake, of course. So thanks again for volunteering to the program, for joining us today for our seminar, and thanks to Rebecca and Colin and Michelle for helping out with the webinar. And we look forward to speaking with you over the weeks and months ahead. Enjoy the coming weekend and the warm summery weather that's it's sure to come. So take care, everybody, and all the best.